in an experiment.
And we don't know yet.
Why is light so far?
Like, it sounds so simple.
They had no idea.
But now the data speaks.
I find this not only refreshing, but at some level astounding.
Nature.
Welcome back to the Nature Podcast. This week, a major step towards a nuclear clock.
And how engineered T cells could improve spinal injury outcomes. I'm Benjamin Thompson.
And I'm Lizzie Gibney. Every clock needs an oscillator to help it keep time. Something that's rhythmic, like a pendulum. And for the best clocks in the world, the pendulum is something that swings very fast indeed. It's a frequency of laser light. To be precise, the exact frequency that will bump or transition an electron up or down an energy level in atoms of strontium. Such atomic clocks are already so accurate that it would take 40 billion years for one to lose or gain a second. But because these clocks are fragile and finicky, keeping them precise takes a mammoth effort. Physicists have long wondered if they could make something more robust, and maybe even more precise. A clock that ticks not using electron transitions, but sturdier transitions of protons and neutrons within the atomic nucleus itself. Now, after 50 years, physicists have nearly done that. They've taken a crystal studded with a rare isotope of thorium and used lasers to nudge a bunch of the element's nuclei into a higher energy state, an important step in the creation of the long hypothesized nuclear clock. And they've measured the frequency of the shift, the clock's tick, with unprecedented precision. Because the speed of this clock's tick depends on fundamental forces of nature being what we think they are, already the setup is being used to hunt for exotic new physics. I spoke to Changquang Zhao from GILA, a research institute in Boulder, Colorado, about the work. I started by asking him about the best clocks we have today. Just how accurate are they?
So the most accurate clock is actually next door to our lab, and they are right now having a systematic accuracy level on the order of 10 to the minus 19.
So if you had two clocks like that, they would agree with each other in terms of the seconds that have passed at the 19th digit?
Yes, exactly. So you have a 19-digit sensor, and then you can use it to measure really a lot of fundamental physics.
And what makes a nuclear clock potentially even better than that kind of atomic clock?
So instead of using electron orbits inside the atoms, which is what atomic clocks use, we actually use the protons and neutrons motion inside the atomic nuclei. And this clock operates at a higher frequency, and this gives us a very high precision. And because we are using the nucleus, which is shielded by the outer electron shells, this actually gives us a much more robust clock oscillator that is less sensitive to external electromagnetic fields, for example.
And so can you just use any nucleus to make a clock, or does it have to be a very particular one?
So right now we can only use thorium-229 isotope because this special unique nucleus has a very low lying energy transition.
So the transition is low enough energy that with some effort you could just about use a laser in the lab to trigger it. What did you need to actually make that happen?
Yeah, so essentially people know this nuclear transition existing for almost 50 years by now, but the transition energy hasn't been measured very precisely until last year. Then we finally have a transition energy uncertainty narrow enough that we can start with our laser search. And we also needed this great crystal material that also took a lot of technical development, where people embed a lot of thorium atoms inside the crystal material to enhance our signal to noise ratio.
So you now have enough thorium-229 in a sample where you hope you can see a signal and you have a good idea of where that frequency is going to be. To make the clock, you need to stimulate that frequency with a laser. How did you find exactly the right energy? Because it has to be precise, doesn't it?
Yes. So there is still a very large frequency range that we're searching for. So we use a device called a frequency comb. It's kind of a special laser where we have a very broad band coverage, but we also have many spectral lines in there. So that we can use all these spectral lines to scan in parallel, so we could get a very large coverage at a very fast speed with a very precise scan steps.
And so rather than kind of scanning through lots of different frequencies. You fire them all out at once in this kind of comb. Would you then see which one matches best with the nuclear transition?
Yes, exactly. So when we first excite the nuclear transition, we actually don't know exactly which is exciting it, but at the end it becomes a math puzzle that we can solve for the actual nuclear transition frequency.
And when you saw it working after all this effort, how did you feel?
I actually felt a little doubtful about the signal because we failed so many times. It was late at night, but we spent the entire night doing all the tests to check if this is actually really the signal that we're looking for, or if it is just some random electronics problems. And finally, we were able to confirm that. So it was like a great relief for me as well.
And how much better do you know the frequency through this method than people have been able to figure out before?
Earlier this year, there's been measurements that already improved on previous measurements by several orders of magnitude. And now today we are doing even better by, I think, another factor of 1,000,000. So we are really resolving the nuclear transition to a much, much more precise value.
You did something else as well. The comb allowed you to kind of hook up the nuclear clock, or the transition that, is the oscillator of a nuclear clock, with one of these atomic clocks, the one that's in the lab next door to you, I think. Why do you do that? What advantage does that bring?
So you always need a synchronization to understand how clocks work. Also, we are more excited about this synchronization between nuclear clock and the atomic clock because it allows us to do more core physics. Because the nuclear clock actually depends on different kind of interactions that goes into protons and neutrons. By comparing our nuclear clock with atomic clock, which are more based on electrons and photons, we can really learn a lot about core new physics.
So this is kind of a way to see what's going on inside the nucleus.
Yes.
And how well do we understand that? Do we think that it's likely to reveal anything?
Yes, so essentially we know our universe has a lot of dark matter, and we don't know how this dark matter interact with our ordinary matter that we're familiar with every day, besides from gravity. And now there are a lot of hypotheses that they might actually perturb the nuclear transition differently from atomic transitions. So now if we see the ratio of the nuclear clock and the atomic clock changing over time, this could be an indication of some dark matter interaction that causes the frequency ratio change.
So if the tick of the nuclear transition changes compared to the atomic clock based on electrons, this could be due to dark matter messing with the forces inside the nucleus. This is like a window into a world we've not been able to probe before.
Yes.
And is what you've built technically a clock? Because I feel like if we say this is a clock, we could get some angry emails because you're not actually using it to keep track of time yet, right?
So it's technically not exactly a clock yet, because first we want to make our laser systems better, or how the, say, crystal temperature changes the clock transition frequencies. And then when we make it a clock, it will be a great clock.
And how good could it be? We know that this transition is a bit more stable because it's inside the nucleus. It's not affected by the same things that affect the electrons. How much better could a nuclear clock be than the best clocks we've got right now?
So one big motivation for this nuclear clock is that we wanted to make it portable, that you can really deploy for field precision metrologies. Now, for these kind of portable clocks, we think we can get 10 to the minus 16. So that's not as good as the best clocks in the lab, but will be outperforming all the portable clocks that we can take out from labs. For the absolute best atomic clocks, we still have a long way to go, but that's because also people have worked on atomic clocks for such a long time. I think we can likely reach 10 to the minus 19 levels, so similar to the existing atomic clocks. And also then in terms of sensitivity to fundamental physics, the nuclear clock actually offers us an unparalleled advantage over atomic clocks. And we are sensitive to different types of new physics, so our sensitivity could even be 10 to the 8, so 100 million times better for certain fundamental physics questions compared to atomic clocks.
And it sounds like there's quite a bit of technical work to turn this into a portable clock or make it as good as the best optical clocks. But the physics can actually start today, can it?
Yes, so there are already theories using our experimental data showing that we can actually be sensitive to dark matter that are as sensitive as existing atomic clocks today. And we're using this resolved quantum structure of this nuclear transition to test our understanding of nuclear structure, how the different nuclear moments behave.
So there's a lot to do already.
Yes.
That was Changquang Zhao from Gila in the US. To read his paper, look out for links in the show notes.
Coming up, how engineered immune cells could help limit damage after spinal injury. Right now, though, it's time for the research highlights with Emily Bates.
A star in a nearby galaxy has been spotted being almost destroyed by a supermassive black hole. Not just once, but twice, and could have another brush with destruction soon. Almost every large galaxy is thought to house a supermassive black hole at its centre. When a star passes too close to such a black hole, it is stripped of material, releasing a burst of light. The star is typically torn apart, but if it survives, the event is called a partial tidal disruption. One star, not far from the Milky Way, endured partial tidal disruptions in both 2022 and 2024. Researchers suggest that it orbits near to its galaxy's supermassive black hole and undergoes a partial tidal disruption on its closest approach. The team hopes to observe a third partial tidal disruption in the next 2 years. To learn more about this unlucky star, head on over to the astrophysical journal Letters. According to a laboratory simulation, bumblebees might lose their sense of smell during heat waves. Researchers subjected bumblebees from 2 separate species to almost 3 hours at 40 degrees Celsius in a climate-controlled chamber. The bees' antennae were then snipped off and attached to a machine blowing floral-scented air. The heat reduced the responsiveness of antennae to floral scents by up to 80% in female worker bees and by up to 50% in males. The findings could mean that as heat waves become more frequent and intense, heat-stressed bumblebees pollinate fewer flowers. A reduction in pollination could, in turn, reduce plant reproduction and hurt bee colonies. Read more at Proceedings of the Royal Society Bee.
Next up on the show, reporter Nick Petrichow has been looking at some research into using the body's immune T cells to help speed up recovery from spinal cord injury. Here's Nick.
When there's an injury to the brain, spinal cord, or anything else that makes up the central nervous system, the CNS, there are some things that researchers understand well and some things that are still a bit perplexing. Let's start with what they do know.
So for spinal cord injury, you damage a particular number of neurons by the primary damage. Those will die quite fast, and their death will trigger a series of events which will continue and kill other neurons which have not been damaged by the primary insult.
This is Jonathan Kipnis, a researcher who works on neurology and immunology. So after getting damaged, the eventual injury will end up much bigger than it started. There's a lot of complex interactions that seem to underlie this, and part of that picture is the immune response. And that's where we start to get into the less well-known territory.
In the central nervous system, the assumption has always been that immune response is detrimental.
Following an injury, immune cells rush to the site, but the effect they're having isn't well understood. It had been thought that inflammation and the actions of immune cells might be causing the further damage. These days, researchers are increasingly understanding that the immune response isn't necessarily a bad thing. In fact, it may be helping in this scenario. But there are still aspects of it that have been a bit contradictory. Take, for example, the actions of T cells.
There are many papers that suggesting that T cells which promote inflammation are the T cells that will be beneficial in CNS injury. And there are other works that say that T cells that suppress inflammation are beneficial. And I think, why can it be both?
Jonathan's point is that the immune response to a CNS injury is very complex and has competing factors. So inflammation may actually be a double-edged sword here. You may want a bit of inflammation to get rid of any bacteria or viruses that have crept in, but you also don't want too much as it might cause further damage. Because T cells have a key role in inflammation, Jonathan wanted to investigate what they're doing at these injury sites. It's possible that they're able to tune the balance of inflammation, and if you could get that right, it may be possible to prevent the secondary damage that occurs when there's a CNS injury. So he turned to mice to investigate. In these model organisms, he was able to damage their spinal cords, collect all the T cells that gathered at the injury, and sequence their individual DNA to see what made them tick. Now, T cells come in several different flavours. Their flavour is based on their receptors, proteins that sit on the surface of the cell that recognise different things. It was the genes encoding these receptors that Jonathan was interested in sequencing.
And then what we see is we have more than one T cell with the same exact T cell receptor. That suggests that the cells are proliferating.
Certain flavours of T cells were rapidly increasing in number at the site of the injury. So Jonathan figured these ones might be playing a key role in preventing secondary injuries. To find out, he took the genetic code for those specific receptors and put them into blank T cells in the lab. With that, he could see what they responded to. It turns out these immune cells seem to recognise parts of the CNS itself, suggesting that they are autoreactive. They target our own tissues, which is generally thought to be a bad thing as it could damage our tissue. But in this case, Jonathan found that they did so only weakly. Still, Jonathan figured that this weak autoimmune reaction could potentially be avoided, so he created a set of T cells based on these receptors that would only work temporarily. It was possible that these temporary T cells could fine-tune the inflammation and stop secondary injuries without damaging the tissues too much. To find out, he injected them into mice with spinal cord injuries to see how it helped them recover.
And what happened is that the recovery of spinal cord injury was substantially improved in response to some T cells, but not to all.
Jonathan used a measure of mouse locomotion to determine how well they were recovering from their injuries. Essentially, if the mice were able to move around better, it implied that they had better recovery. Not all the T cells that were particularly numerous at the injury site were beneficial here. Some didn't seem to have much of an effect on the mice, but some did, scoring higher on the locomotion measure. Now, according to Jonathan, it doesn't seem like the mice's original injury is healing any better. What seems to be happening is the injection of T cells is preventing the spread of damage. Preventing this spread leads to a smaller overall injury, helping the mice to heal more quickly. The way this appears to work is through T cells tuning the inflammation around the injury by using a molecule known as interferon gamma.
So yes, we believe that it's a T cells that controlling local inflammatory cells, at least in part through interferon gamma. I'm not saying this is the only molecule, the most important. And even if it was, you cannot inject interferon gamma because the beauty of T cells is mini factories, which are mobile mini factories, right? So they arrive, they sense the environment, and then release those molecules. Plus, maybe they're releasing 25 or 250 different molecules.
When Jonathan blocked the T cells' ability to produce interferon gamma, they weren't as effective at stopping the secondary injury. However, he thinks it's unlikely that simply injecting interferon gamma would be a way to help the mice heal. He also tried injecting a general cocktail T cells that should be relevant to clearing up the injury, but this doesn't work either. That means to reproduce the same effects, you would need to rely on the specific T cells that gathered at the site, which means collecting them from the injury site, sequencing them, and then making the temporary versions again, all of which takes time, during which an injury can spread. But there's also the question of whether the mice results are relevant to humans. Jonathan and his colleagues have been looking to see if the T-cell proliferation they saw in mice has parallels in humans who've experienced CNS injury.
The pattern of T-cell proliferation and how they look at different days and the types of T-cells that we see proliferating gives us a lot of confidence that these are the ones that we also see in mice. So now, of course, the next step is to develop method to do what we're doing in mice, doing it in humans.
That's not all he's thinking of for humans, though. In CNS injuries, it's a bit of a race against time to stop the nerve damage spreading in a matter of weeks. But there are many conditions that are neurodegenerative where damage can spread over years. So Jonathan wonders if there's something they can do here.
For example, ALS, where you see a death of motor neurons in spinal cord. Of course, it goes over years, right, the disease progression. And so there we have time. We can take the cells out. We can understand specificity. So will our method work in ALS? And while that's not published, in this paper, but we do have some very promising results with ALS. So, maybe we could start with neurodegenerative conditions, which are more chronic. I really believe, the T-cell therapy revolutionized immunotherapy for cancer. I think CAR T-cells are very, very promising. And so the question is, can we build something similar also for neurodegenerative diseases? And that's a big question. I don't have an answer of yes, but I really hope that the answer will be yes.
That was Jonathan Kipnis from Washington University in St. Louis in the US. For more on that story, check out the show notes for some links.
Finally on the show, it's time for the briefing chat, where we discuss a couple of articles that have been highlighted in the Nature briefing. Ben, what have you got for me today?
Well, I've got a fascinating story I read about in Nature, and it's about the race to find and save some fossils in Brazil that have been threatened by incredibly intense rain.
And there have been some major floods, as I understand it.
Yeah, particularly in the south of Brazil. And this story focuses on the state of Rio Grande do Sul. Now, this area saw some devastating floods in May this year, and the state's capital, which is the city of Porto Alegre, saw almost half of what it would annually in like a month, right? So 66 centimeters. And this story is looking at how rain can kind of be a friend or foe to paleontologists because, of course, it can cause landslides and erode away soil and rock, that kind of thing.
So it can both reveal potentially new fossils and new finds, but also destroy what's there at the moment.
Basically, that's exactly right. And that's what this story has looked at. And so particularly this story looks at some of the findings that a group of researchers have made in this area 280 kilometers away from Porto Alegre. It's quite fertile ground for fossil hunters. Now, in the weeks following the floods, the river system in the area obviously overflowed. And after some of this water had subsided, the team uncovered this amazing fossil at 2.5 meters long of a carnivorous bipedal dinosaur belonging to the Herrerasaurid family.
What kind of a beast was that?
This is potentially, one of the first top predators among the dinosaurs. So, kind of a big deal in dinosaur terms. And it lived in the Triassic period, so 250 to 200 million years ago. And it kind of emerged and vanished in this time. And the researchers are quite excited about this. They think it could be a really complete skeleton. Now, it hasn't been peer-reviewed, but it seems that it may be among one of the oldest dinosaur fossils in the world, and they're taking a slab of it back to the lab to try and really carefully uncover it and make sure they can have a look and see what they've found.
Wow, because I was going to say, presumably these awful floods have these little tiny silver linings in terms of science, and it gets uncovered, but then you might be in a race against time to actually haul it out of the situation it's in, because there's going to be ongoing flooding, there's going to be potentially deteriorating weather. How do they deal with that?
Yeah, well, that's absolutely right. I think a race is the right word. In this case, the researchers say that this has kind of been a difficult discovery to celebrate because these floods, they've affected, 2 million plus people. There have been many deaths and many missing people. So you can see that their excitement has been tempered. But as you say there is this knock-on effect as well. And there have been more intermittent rains subsequently. And I think I said that rain could be friend or foe. And in terms of the foe, it really can damage smaller fossils, these ones that maybe aren't quite so box office, right? Everyone loves the big bipedal carnivores, but there's a lot of diversity in the smaller species. And certain bones, like ear bones of fossilized animals, are really, really important in working out how evolution followed and where they lived. And these ear bones, as we've covered in the podcast, can be, like millimeters in length, like really, really small. And obviously they can be damaged quite easily by heavy rain and floodwaters and what have you. And there have been some of these smaller fossils that have been found. The team recently uncovered the skull of a baby rhynchosaur. Now, this is a parrot-beaked herbivorous reptile, right? Like maybe about a meter long when it was fully grown. These dominated the Earth during the Triassic period. As I say, not particularly box office. They're found all over the place. But they're actually quite useful because they can act as markers for when the Triassic period was. And if you find a bunch of these, well, there may be something more exciting to find there as well. So it's important that researchers can get access to these sites.
And is it all the same site near Porto Alegre, or are they spread across Brazil?
In this instance, it's this particular site in Rio Grande do Sul, so this state right at the southern tip. And there's like 29 different excavation sites. 21 have been accessible since the flooding. 4 are still underwater, either completely or partially. But the team, as I say, have found a bunch of different things. So they found pieces from at least 35 other animals. And I think interesting problem to solve is what to do with them, right? Because they don't have a museum, and it's been quite rich pickings at the moment. So it's an interesting one to find out how they can use this knowledge and where they can display it. And as I say, it's a really interesting story, and I'll put a link to that in the show notes. But let's move on in today's briefing chat. Lizzie, what have you got this week?
So all of your problems are solved, dear listener. We have an AI scientist. Hooray, everyone can go home.
Finally.
So this is a story in Nature, and it's from a preprint. Now, there'll be a lot of skepticism about this, and justifiably so. What's happened here is that some machine learning researchers in academia and with a company called Sakana AI, they've used AI to do what they call the full cycle of research. So from the literature review stage to kind of figuring out what is known about a problem, to making a hypothesis of how you can go beyond that, trying to solve the solutions in different ways, and then ultimately writing a paper about what they found. That was all done full cycle using AI. So that's the bit that's really new, because we have had people try and automate different bits of the research process before, but this seems to really be the first to do the full circle. Now, caveats up top, because I can see that you're dying to add some. This is only in machine learning, so something that can be done on a computer, on paper. It's not lab or field work, which is obviously where a huge amount of the scientific process happens.
Yeah, I mean, I think the end bit of like, put all these chemicals together and work out if you can come up with a new enzyme or a new catalyst is something that's been done a lot. But this one is the sort of tapping a pencil against your chin and leaning back in your chair in the office stage of science, as I remember it, that this is maybe trying to emulate to an extent.
Yeah, so there's been a robot chemist, for instance, which I think might be what you're referring to before. And, you know, that is very hard for many different reasons, like robotics is a whole different beast that is incredibly hard to do and to link up with AI. This is indeed the bit which maybe some people don't like about the process, you know, the literature review in particular. So the way that they've done it is they've used a large language model as a base. We've spoken about those many times before. They took as a template a paper that describes one particular machine learning algorithm, and then they got this large language model to kind of evolve from that. So they use this kind of evolutionary method to try out random changes that would make the algorithm better, and then try those out, pick the ones that work better, and ultimately improve upon this algorithm. It did experiments using that algorithm. It measured the performance. It then wrote a paper, and then it kind of almost even peer-reviewed itself. The authors are very aware that, like, the paper that is produced is a very incremental advance, right? A commenter said that they would have rejected the paper if it came to their journal. And it's learning just from what's out there in research papers. It's not chatting by the water cooler. It's not going to a conference. So there's an argument to say that it's not likely to make those kind of really innovative scientific leaps. And in my head, it's not an AI scientist, it's kind of an AI that makes new papers. It's kind of almost like looking for things that could be novel enough to be a new paper, but that's not really what science is about. We write the papers for good reasons, but science isn't about producing more papers.
Yeah, the enterprise of science, if we can call it that, is so much more. Now, often when we have stories like this, one of the questions is this going to do scientists out of a job, right? And usually the answer is no, or at least not yet in some instances.
I would still very much think that that's the case. A few positives about this paper and the things that it can do. Well, for a start, the paper is all open. So, you know, they're not being shady about it. They're not saying we are taking over your job. You know, they're saying, look, hey, this is what we've tried to do. Come and have a look. See if you think it's any good. See how you could use it, how we could explore it. So in that way, it's very kind of collaborative. And there's a sense that it is just worth exploring, because whilst many people would not like an AI scientist to take their job, very understandably, there are parts of the process that maybe we would like automated, that maybe are repetitive, that take time that could be usefully spent on something else. So there is this question in the scientific community of what bits of the job could be done by an AI. And this is a really useful entry into that.
And is AI as a sector a fruitful one, getting an AI to work on AI problems? Is that useful? We've talked about chemistry there. Is that more difficult for it to get into?
Anything that involves a lab will take a completely different kind of AI and process to make that work. But in any case, it looks like, you know, this is, as I said, a large language model with some other algorithms on top. It might take a completely different kind of artificial intelligence to do different parts of this process, something that perhaps that has logic rules built in. It's called symbolic AI. That's had more success with solving mathematical problems recently. We talk a lot about the limitations of large language models. But hey, there probably is somewhere that AI will fit into science. So why not have a go at doing it in this paper. So it's just a preprint at the moment. Let's see what happens when the paper gets peer reviewed. But I think it's something that's worth exploring.
AI getting involved in research is something that you've been onto the show to talk about a lot and is on the radar, I guess, for a lot of people, for better or worse. What's been the reaction to this work in the community?
It's been pretty mixed, I would say. A good quote that's in the story that kind of sums it up is someone saying, people will say that they have like 100 ideas that they don't have time to look into. Get the AI scientists to do that. So that seems like a good starting place and a good summary of what people think about this.
Sort of on your little side projects that are just in the corner.
You've got an inkling, you've got an idea. Hey, AI, go, have a look at this, see what you come up with. Maybe it comes up with something good, maybe it doesn't. Maybe that's a starting point to launch a full research project. Who knows?
Well, I have a sneaking suspicion that that's not going to be the last AI scientist story we talk about in the next days, weeks, months, and years, however long it takes. But let's leave it there for this week's Briefing Chat. And listeners, for more on those stories and where you can sign up to the Nature Briefing to get more like them delivered directly to your inbox, check out the show notes for some links.
That's all for this week. As always, you can keep in touch with us on X, we're at Nature Podcast, or you can send an email to podcast@nature.com. And if you fancy leaving us a review, you can do so wherever you get your podcasts. I'm Lizzie Gibney.
And I'm Benjamin Thompson. Thanks for listening.